Hello everyone. In this video, I am going to tell you about the dysplastic features. The term dysplasia was first introduced by Regan in 1958 in relation to the cells exfoliated from lesions of the uterine cervix. The word dysplasia is derived from dys, which means abnormal or bad, and plasia, which means growth. So dysplasia means abnormal or bad growth. Dysplasia can be defined as a precancerous lesion of stratified squamous epithelium characterized by cellular atypia and loss of normal maturation and stratification, which is short of carcinoma in situ. WHO in 1978 described 12 dysplastic features which can be used to grade the severity of dysplasia of various precancerous lesions. And these dysplastic features include enlarged nuclei in cells, large and prominent nucleoli, increased nuclear-to-cytoplasmic ratio, hyperchromatic nuclei, that is, the cell becomes dark in color. Pleomorphic nuclei and cell, which means that the shape of the nuclei and cells become atypical. Dyskeratosis, which means abnormal keratinization, and single cell keratinization may also be seen. Increased mitotic activity, abnormal mitotic figures, which means that a single cell can be seen dividing into 3 or more cells. Bulbous or teardrop-shaped rete-ridges, loss of polarity, keratin or epithelial pearls, and loss of typical epithelial cell cohesiveness. Now let's see an animation detailing these dysplastic features. Because of the loss of normal regulation in a cell, the genetic material starts to increase rapidly within the affected cell, and this increase in the genetic material leads to the enlargement of nuclei which contains more DNA as compared to the normal cell, and due to this the cell and the nucleus, they both increase in size, though the increase in the size of the nucleus is more as compared to the increase in the size of the cell. And this excess abnormal genetic material causes abnormal mitotic division. And because of this abnormal mitotic division, the cell, instead of dividing into 2 daughter cells, it may divide into 3 or 4 daughter cells. As you can see in this animation, the nuclei of the affected cell is dividing into 3 parts. Due to expression of the abnormal genetic material, the shape and size of the cell and the nucleus changes, which is known as pleomorphic cell and nuclear form. Also, because of the abnormal genetic material, the rate of mitosis increases, and this leads to the rapid proliferation of the abnormal cells. And because of this rapid proliferation, the rete-ridges, as you can see here, starts to expand because of the pressure exerted by these abnormal cells. And since the genetic material is abnormal, the normal characteristic features of the cells are also lost, and because of this, the intercellular adhesion becomes weak and at times it is lost. And because of this loss of the cellular adhesion, the cells become free and they are free to move either in the underlying connective tissue or within the epithelium. The next dysplastic feature is the loss of polarity. And before we know what is the loss of polarity, we must know what is polarity. So a normal epithelial cell maintains a typical relationship with the basement membrane and the surface of the epithelium. What it means is that the cell in the stratum basale proliferates and differentiates into stratum spinosum cells, and these stratum spinosum cells, when they move towards the surface, they further differentiate into the stratum granulosum cell, and these stratum granulosum cell, they keep on moving towards the surface and they differentiate into the stratum corneum cells, that is the keratin layer which is seen on the surface of the epithelium, and from there it exfoliates. So this is the normal polarity of a normal cell. But in case of an abnormal cell, this normal polarity is lost, and because of the loss of normal polarity, this abnormal cell moves randomly within the epithelium. It matures within the epithelium, which leads to the formation of intracellular keratin, and this intracellular keratin is also known as Dyskeratosis. Similarly, a lot of abnormal epithelial cells would move randomly within the epithelium, and these abnormal cells accumulate in a centrifugal pattern while they are maturing and keratinizing. And because of this simultaneous keratinization of adjacent abnormal epithelial cells, the structure that forms is known as keratin pearl. So these are the different dysplastic features as described by WHO in 1978. Now let's see how we grade different lesions based on the presence of these dysplastic features. So the grading can be done as mild, moderate and severe. In case of mild dysplasia, the dysplastic features are limited to within the basal third of the epithelial thickness. In case of moderate dysplasia, the dysplastic features are limited to the basal 2/3 of the epithelium, and in case of severe dysplasia, the dysplastic features involve more than 2/3 of the epithelium thickness. At times, this dysplastic features may reach even to the superficial layers. That's all friends. I hope you like this video, and if you find this video helpful, please like, comment and share, and please don't forget to subscribe. Thank you.